Hello again. Welcome back to Botany 101, General Botany. My name is Angela Nishimoto, and I'm sort of like your guide for the tour, okay? So today, our subject is going to be classical genetics, and this is session 14. Okay, so we're going to get into classical genetics. Okay, our objectives for today include the basic principles of inheritance, Mendelian genetics, and other patterns of inheritance. It can get kind of complicated, okay? So first of all, remember, this is something that we talked about before. This here, what you see on the screen here, is a leaf of kalanchoe or kalanchoe. People pronounce it different ways. Some people call it air plant. And when you see right here on the margins of the leaf, right at the very edge, here you have little keiki. So these are actually plants, little baby plants growing from the leaf. And this is an example of asexual reproduction. You remember that asexual reproduction happens when you have offspring plants growing from either the root or the stem or the leaf. Okay, and so these little baby plants here will be genetically identical to its parent plant. Okay, we'll have the same genes, the same genetic makeup. All right? Now, of course, too, the topic we're going to study today has to do with genetics. And what is genetics? It is the study of inheritance. Okay? So what happens? This is like a variation of that alternation of generation that we saw before. Remember that plants undergo alternation of generation. So starting here at fertilization, okay, where you have sperm and egg coming together, right? You have fertilization when sperm and egg fuse, and then after that you have the growth of the sporophyte. The sporophyte is actually usually the plant in a diploid state. Okay, so here you actually see this cell here and this organism that's represented by this rectangle, okay, has two sets of chromosomes, all right? It has 1, 2 long ones and 1, 2 short ones. One of the short ones and one of the long ones of the chromosomes would have come from mom. The other short one and the other long one would have come from dad, okay, from the sperm. So we have egg and sperm. So when we actually do have fertilization in us humans, as well as how it happens in plant, our chromosomes actually do get passed on literally to our offspring, okay? The chromosomes that we inherited from our moms and dads. Another thing that happens too is that this sporophyte, you know, this diploid sporophyte will undergo meiosis in its sex organs, producing 1, 2, 3, 4. Okay, generally 4 cells result from meiosis. Each of these cells different from each other, and each of them different from their parent cell. Because for one thing, the resulting gametes or sex cells or sperm or eggs are haploid. You see, it only has 1, 2. It has a haploid number of 2, whereas this cell here has a diploid number of 4. 2 sets of chromosomes, whereas this cell and this cell, okay, has only 1 set. Okay. Alrighty, let's see. So generally inheritance, so these are the traits that you as a person manifest, whether you have dark hair or whether you have dark skin or whether you have green eyes or any combination, you know, of coloration and structure, whether you're tall or short, you know, all this kind of stuff. These are all traits that you have inherited from your parents that are coded for in your DNA, okay? So your traits actually pass to you from your parents, all right? Okay? Alrighty, so traits then, okay, so here, okay, we have a bee. So here is showing sexual reproduction in a flower. This bee acting as a vector, transferring pollen from one flower into another flower, perhaps, okay? Another thing too is that generally another example of sexual selection and inheritance is artificial selection. Agriculturists, you know, people who are growing crop plants. Here's a picture of a farm. Okay, this looks like a little farm here, and they have their bees, honey bees, in these little boxes. And of course, the bees in this instance are actually doing the cross-pollination. Okay. So examples of these include domesticated plants, of course, because domesticated plants are just plants that we humans have grown, selected for, etc. Okay, so we have wheat in one example, giving us wheat berries. Okay, this is great. Look at that. Okay, then we have corn. You can see the tassel at the silk at the top. Okay, the tassel would be higher. And you have the male and female flowers of corn. And here is the abundance of corn. Look at that. Doesn't that make you feel hungry? Yeah. And then we have rice, and here's a person in a rice paddy, looks like in Asia someplace, with little rice seedlings. And of course she also, and here are the rice grains, right? So if that doesn't make you hungry, you know, that actually should. Yes? So you have these domesticated plants, the big three grains, wheat, corn, and rice. We also have things like locally here, which supported and maintained the lives of, you know, hundreds of thousands of native people of Hawaii, including the kalo. Okay, so this is the kalo plant that sustained the life, you know, and sort of like the whole culture, partly. All right, here's some kalo plants. It looks like a dryland kind of taro situation. Okay, and here are the corms. You can see in this particular variety that you have this really nice kind of purplish grayish color. So usually when you buy poi at the supermarket, because almost nobody makes their own poi or pounds their own poi anymore, but still it has that really nice kind of purplish coloration in it. And when you go and buy poi rolls or taro rolls or even taro chips, usually it has that nice bluish purple kind of tint to it. Very, very good. And this looks like, this is a photograph taken by Dr. Carr, you know, Dr. Gerald Carr, of late of the University of Hawaii at Manoa, Botany Department. And this looks like the Hanalei Valley. You see acres of kalo plants, yeah, in a lo'i kalo. And then another kind of domesticated plant, which is kind of famous, is like cabbage, okay, also broccoli, and also cauliflower. Okay, take a look at these, and also kale. I think this is called kale. And we have kohlrabi. That's magnificent. Look at those guys. And then you have Brussels sprouts. Guess what? All of those different kind of plants are all this one species. Okay, Brassica oleracea. All are very different from each other, and they are actually the result of human selection over hundreds of years. It's only taken several hundred years for humans to actually be able to develop all these different takes. These are all the same species. They're all variations on the same theme, but they're all the same species. Isn't that amazing? Okay. Now, of course, you know, so genes, okay, are what people study in genetics, okay, genes, which we call genes now. And what are genes? Genes are actually on DNA, and DNA, of course, is a component of the structures known as chromosomes, remember? So here's a double helix of DNA molecule. You can see that you have one strand, you have a second strand. They're like a rope ladder that's been twisted, okay? So what happens is that some people say that DNA is actually two molecules, hydrogen bonded at their center. You see here and here, okay, where you have the nitrogenous bases, you have hydrogen bonding there between one strand and the other strand in DNA, okay? And of course, here, this is a chromosome that has undergone duplication, so you can see that this single chromosome has an exact copy on this side. So if you recall, this is called a chromatid. When you have these chromosomes that have been copied, so you have one chromatid, you have another chromatid on this side. Secondly, you also have the part where the two chromatids are joined together, the two sister chromatids being genetically identical, and they're joined together at a part called the centromere. Okay? In this chromosome, you have a short arm, and you also have a long arm of the chromosome. Okay? So this is like a typical chromosome, okay, when it's ready to undergo either mitosis or meiosis. All right? So genes are a unit of inheritance. So what it is, a gene is actually a stretch along DNA, okay? And this is a good illustration because it shows that this DNA, packaged with proteins, are actually coiled, super packed. So actually, if you actually took the DNA from a single cell, you could actually stretch it about 2 or 3 feet, you know, from one cell, all the DNA in the chromosomes from one cell. So what it is, it's a miracle of packaging. This is good too because what you see here is that this gene, okay, this gene that is that's going to be transcribed and later translated has non-coding regions called introns, right here, I'm sorry, introns. This area here is a part of the DNA that doesn't code for a product, whereas these exons will actually be joined together, spliced together later on, we'll get into that later, but just remember for this part that introns are actually non-coding regions of the DNA. It's sort of like a problem that scientists are working on now to figure out what the introns are doing and what the proteins in a chromosome are doing there. okay? Because nature doesn't waste anything. We don't have anything in nature that is a waste, okay? So these genes code for a polypeptide, okay? So each gene will code for one polypeptide, okay? What is a polypeptide? A polypeptide is a string. If you recall, when we talked way back about chemistry, we have a whole string of amino acids, okay, that make up a polypeptide. Okay, poly means many, peptide refers to units in proteins, okay? So these polypeptides, okay, sometimes you can have either a third level of protein structure to fourth level of protein structure. And anyway, so these polypeptides will control all the aspect of a particular organism, whether plant, animal, fungus, bacterium, protist, okay? It controls the metabolism, all the different chemical reactions that go on in the plant as well as in other organisms as well. These polypeptides also control what size the organism is going to grow to, what color various parts of the organism are going to be, the development, how quickly it matures or how slowly it matures, how quickly it senesces or ages or how slowly it senesces, okay? Just like us, if we have really good genes, generally we tend to look really, we tend to look very young when we're older. If you have good genes, that can happen. Of course, the environment also has a part to play in this. If you have really good genes and in your family you have a lot of good health, your older relatives or mostly are still around, even if they might be in their 60s or 70s or 80s and they're healthy, that's good. But of course, your environment also has an impact on how long you live and the various various illnesses you don't get, as well as some illnesses that you do get, okay? Another thing that these polypeptides will code for is reproduction, the timing of it, when an organism becomes sexually mature, how many offspring possible can they get, even some phenomena such as twinning. When a mother has twins, identical twins, that too has a little bit of a predilection depending on your genetic makeup. So all your traits, all the traits of you, all the traits of all plants are actually controlled by the genes in the chromosome, in the nucleus, right? Sort of nested inside each other, sort of like those Russian dolls, okay? Anyway, so genes coded for the height of this huge cactus here. Look at these little people, yeah? And I think that this woman is probably at least 5 feet tall, maybe a little taller, and then this little girl, so look how big. So here's a cactus, so we're just going to look at a little bit of some variation just in plant, and this is just the plant kingdom. So this is a cactus, of course. Look how big it is. Isn't it magnificent? And then here you have a nice photograph, also taken by Dr. Carr, of, I think this is called, let's see, it's related to the native vili vili. They call it Indian coral tree, I think is what it's called. And right now, at this time in the season, sometime in early summer, late summer, you can see a lot of these trees blooming. Sometimes maybe they call it the flame tree too. Yeah, I think they also call it Indian coral tree, or Erythrina, yeah? And then we also have bamboo. So these are all members of the plant kingdom. Not only are they members of the plant kingdom, but they're also flowering plants. They're all, angiosperms. Okay, so here we have a nice photograph of bamboo. To me, it looks like it might have been taken at the Pagoda restaurant. It sort of looks like that. I don't know if it has been, but and we also have something like this, which is the white ginger. Okay, yeah, Hiddekium, yeah. Okay, so a locus, okay, is actually the location of a gene. So we have the same image here. So this can actually stand for a gene here, of this length of DNA from one part to the next part, okay? So the location of that is called a locus, okay? And recall too, you have a single chromosome here, you have the long arm, you have the short arm, and we also have these banding patterns, because one of the things to remember is that chromosomes will stain. Yeah, that's why they call them chromosomes. Because generally, if you don't stain them, you can't really see very much what's going on. Another thing is that when you do stain them, they'll oftentimes have characteristic banding patterns, depending on what chromosome it is in the genome of the organism you're looking at. Okay? So, generally, gametes, remember, egg and sperm, whether of animal or whether of plant, gametes have only one set of chromosomes. They're haploid. right? They have a haploid number of chromosomes. Whereas when you have fertilization, you have one set of chromosomes in the sperm, one set of chromosomes in the egg. When they come together, the new individual will have two sets of chromosomes, the diploid number. Okay, one set from mom, one set from dad. Okay? And another thing too is that we also oftentimes have these homologous chromosomes. So what they are is that generally an individual person or plant will generally have two sets of genes. What you see here on the screen here is what they call a karyotype. Okay, what they've done is that they've taken cells from a human being. This is a human being's karyotype, and they actually squash the cells, you know, so that the chromosomes could break out of the nucleus and out of the cell. And then what they did is they made the slide really flattened it as much as they could, took an image of all the chromosomes, you know, in the squash, and then what they did is they computerized it. They took a picture of it and then moved it around. Okay, so here you have the homologous pair of chromosome 1. Okay, homologous pair of chromosome 2, and etc. So this is a human, you can tell, partly because this person has 22 sets of autosomes. That means that this person has 44 autosomes plus one set of chromosomes. And if you remember, we human beings have a set of 46 chromosomes, just as long as we don't have any kind of chromosomal abnormality. Another thing is you can tell this is a man because his sex chromosomes, he has one X chromosome from his mom and his Y chromosome is from his father, his dad. Okay? So this person, of course, is a normal person with 22 sets of autosomes, chromosomes 1 through 22, and then one set of sex chromosomes. Okay? In a case when you have a chromosomal abnormality sometimes, such as they have something called trisomy 21, trisomy 21 means that somehow there's like a mistake during meiosis, okay, when the gametes are produced, and what happens is that the person who has this syndrome, okay, will have 3 sets of chromosome 21. That is actually not that bad of a chromosomal abnormality. If people usually have unusual number of chromosomes, usually the baby is spontaneously miscarried, okay, or spontaneously aborted. Usually the baby doesn't survive to term. But with trisomy 21, okay, it's actually an abnormality that isn't that harsh on the organism, and it results in the person who has it in what we call Down syndrome, what long time ago people used to call Mongolism. Yeah. Okay, so that is a possibility. Okay, there's usually they actually calculate what percentage of births, you know, at a particular time will account for that. So they say as a woman ages, after a woman is 35, as long as she's older than that, usually a woman who gets pregnant after 35, they usually encourage her to have either amniocentesis or chorionic villi sampling. And what that's doing is to check to make sure that the baby is okay, you know, and then if it isn't, then the doctor has to tell the couple, and they have to kind of try to make some kind of decision, okay, because very serious things, okay, sometimes not very pleasant, sometimes sad. Okay, here, and then what happens is that homologous chromosomes, so the individual has 2 sets of genes, okay, and generally alternate forms of a gene are called alleles, okay, alleles. So in one instance, you can have, in a particular kind of plant, like a pea plant, you can have purple flower color, or you could have white flower color. So the gene that would code for this would be a gene for flower color. And the alternate form of the gene, the alleles for this particular kind of flower color would be an allele for purple flower color or an allele for white flower color. Okay? Another thing too, you have these two terminology, homozygous. Homozygous means that the 2 sets of chromosomes, the 2 genes, the 2 alleles will be the same in the individual, such as you could have an individual that homozygous for purple flower color. That means both alleles on the genes, in the genes are for purple flower color, or alternately you could have homozygous for white flower color. Okay? Heterozygous means that you have different alleles. Say, for example, you could have one allele for purple flower color, the other allele for white flower color. Okay? And in terms of pea plants, we're going to find out later what you can expect from that. Okay? Now, the person who is known as the father of genetics, his name was Gregor Mendel. Okay? He did his work in the 1800s. Very, very, very smart man. Very brilliant. Okay? And he's what we call the father of genetics, which is kind of interesting because he was actually an abbot. You know, he was a religious person. He was a monk and a priest. Okay, at the time he did his work, scientists did not know that what nucleic acids were. They hadn't been invented. Another thing is that people didn't know what chromosomes were either. Nobody knew that there were chromosomes. Guys, so his accomplishments, Gregor Mendel's accomplishments, are remarkable, literally remarkable. He pretty much worked almost in a vacuum, okay? Brilliant fellow. Another thing too is the time when he did his work, mitosis and meiosis, the two processes that we studied earlier, were not known. Since people didn't know about chromosomes, they wouldn't have been able to know about mitosis or meiosis either. So he pretty much is working in isolation, okay? So his background, okay? He was a monk, okay, out at Brunn, which I think is now in the Czech Republic. And he also had an abbot, or he had a patron, a senior priest, who allowed him to do a lot of studying. So this monastery that he was at in Brunn was like a place where a lot of science was going on. And he was sent to study at the University of Vienna, which is like a real, old, honored kind of place to study at. You know, probably one of the best universities in the world. Okay, University of Vienna. Okay. Vienna at one point was like a culture center in Europe. And he was very heavy into both biology and physics as well as mathematics. Okay, he had a lot of friends who were physicists. He had a lot of friends that were mathematicians. And here, this is a plate of Mendel. And you can see that it honors him, Gregor Johann Mendel. I think he took the name Gregor as his name as being a priest. And sometimes when people go into the religious life, they take a religious name, yeah? So he actually, I think his name was actually Johann, but he took the name Gregor, I believe. Okay. Another thing too, maybe partly due to the fact that he had all this exposure to physical sciences as well as mathematics, he was the first person in biology to take a quantitative approach. He actually took records, he kept records, and he counted things and measured in that way. Amazing. He's the first biologist or somebody working in biology, working with living things, to take a quantitative approach. Very exciting. Okay. So he actually did his work and he published in 1866, but his work was ignored just because people didn't understand what he was doing. His work was ignored and neglected until about the early 1900s, and then he was sort of rediscovered. Okay, he actually died in obscurity. He wasn't necessarily poor or a pauper or anything like that, but he didn't get the credit for the work that he did during his lifetime. But that's okay. You know, one of the things you'll find out sometimes is that it's not really important who does the work, but the more important thing is actually that the work gets done. That's the most important thing, you know? Okay. So what did he study? He studied something called the garden pea. He had either the luck or the insight to choose the correct organism to do his work with. Okay, so this is a pea plant, garden pea. For one thing, it has really good qualities. For one thing, it's very easy to grow. You can actually have, several generations within a couple of years. You can grow them, they grow into maturity within a year, they flower and fruit, they're easy to grow. Another thing too is that he used true breeding varieties. What does that mean? That means, for example, that he might have a plant that's true breeding for purple flower color. That means that if you self it or you have pollen from one flower fertilizing the same flower, okay, all the offspring are going to be purple, generation after generation. You just keep selfing. That's what they call self-pollination, until you have these true breeding varieties where you have pure for purple flower color, okay, or pure for white flower color. So he used true breeding varieties. And another thing about these garden peas is that they tend to be self-pollinating because the flower itself tends to stay closed. So the only way they can do sexual reproduction is to self-pollinate. Isn't that amazing? Another thing that he did too, this isn't the pea plant, this is actually just some other plant, and we chose this because it shows, a bee doing pollination. With the pea plant, you can also do cross-pollination. If you sort of dissect the plant, you cut off, as part of the structure so that you can be sure that the pollen from one flower doesn't actually pollinate itself. So what you do is you'd actually take a pair of scissors or a razor blade and cut off the anthers to make sure that the flower doesn't self. So what you could do is you could take, say, pollen from a white flower, put it on the stigma. Remember the stigma style and ovary? Would actually put it on the stigma of a white flower, purple flower. You might cross-pollinate that way. So he did reciprocal cross. He put the pollen of a white flower on the stigma of a purple flower. He took pollen from a purple flower, put it onto the stigma of a white flower. So he did cross-pollination. Okay? And another thing too is that these garden peas also generally have 7 pairs of opposing traits. They have 7 easily, identified traits. For one thing, you had some that were tall, okay, and tall was actually what we're going to find out dominant to dwarf plants. So you had tall plants and you had small plants or dwarf plants. You had some with yellow seeds, okay, you had some with green seeds. So generally you didn't have yellow-green, you didn't have medium size. You had either yellow or you had green for purebreds. Some green peas, here's some yellow dried peas. Okay, so when winter's around, isn't a split pea soup a very comforting thing? Yeah? You can also eat hot cereals in the morning, oatmeal or cream of wheat or something. Yeah? Doesn't that sound good? Okay, so we also had smooth versus wrinkled seeds in terms of what the seed looked like, this wrinkling or smoothness of the texture of it. Other traits included green fruits versus yellow fruits, green being what we're going to find later on is dominant to yellow. We had inflated or swollen fruits versus kind of shriveled looking constricted fruits. And you had purple flowers versus white flowers. And then lastly, you had lateral inflorescences. You remember what inflorescences are? They're actually inflorescences that come off the side versus terminal inflorescences. And the terminal ones actually come from the top of the stem. Okay, and here they are. Isn't this magnificent? Look at this. So you have round seeds, round seeds, or wrinkled seeds, or smooth seeds and wrinkled seeds. You had yellow seeds and green seeds. You had white flower color, purple flower color, inflated pods, constricted pods, yellow fruit, green fruit. You had axial flowers. Okay, you had terminal flowers, you had tall plants, and you had dwarf plants. So these traits actually were discrete traits. You didn't have intermediate forms. So he managed to choose the right organisms that had these characteristics that really showed either or. You didn't have anything in between for these. Okay? So this is how he actually did his work. Okay? Another thing that he did is that when he made his crosses, he actually counted what he came up with and he recorded them. Okay, if you were to look in your textbook in the genetics chapter, and there's a table in here, that's table 7.1, and it says, the title is Mendel's Results from Monohybrid Crosses. If you look all the way over to the right-hand side, you'll see that he's actually counted them, well, what kind of seeds he got, and seeds and flower color and all that kind of stuff, and the ratios. Okay, we're going to get into that in a little bit. Okay, so he recorded the types of offspring he got and numbers of offspring that he got. Okay, and this is exciting because it was the first time that in biology data was quantified. He actually counted and recorded and actually did this amazing experiments, Okay. So monohybrid crosses. Okay, so we're going to go into a little bit of doing a monohybrid cross. First of all, we have to be acquainted with some symbols. For one thing, P in this case stands for parental generation. So you can call something P generation or parental generation. Okay. We also have the F1. The F1 is the first filial generation. So this is what you have when you do a hybrid cross, when you have, say, one parent that's purebred for purple flower color, the other purebred for white flower color. You cross these two traits, and then what you get in the first generation, that's called the first filial generation. Okay? The F2 is also known as the second filial generation. That's when you take two F1 plants, okay, two F1 monohybrid plants, and you cross them either to each other or you self one of the plants. And what you get in the F2, okay, that's what you'll get, okay? And so we'll get into that in just a second. Another thing about Mendel's hybrids is that you had generally, when you made a cross, you had one trait expressed, one trait not expressed. So that's what we call dominant recessive crosses, okay? So here, here's actually showing this is representing a white flower or a white individual. This is representing a red individual or a red flower, okay? So the symbols here, not exactly what I want you folks to do, but it does, give you an idea. So when this organism, this one here, okay, makes its gametes, since it's purebred for white, the only kind of gametes or only kind of alleles it can give to its offspring plants are white alleles, right? This one here is red, and it's a purebred for red. And so generally you have only two possibilities when it undergoes meiosis. You can either get red or red, okay? So when you actually cross these two, you have maybe the pollen from one going on to the stigma of the other, or vice versa, okay? And what you have is that in this first F1 generation, the first filial generation, you'll have all heterozygotes, right? Remember heterozygotes? That means they have one allele from one parent, a different allele from another parent. So you have hetero. Hetero means different, okay? It has one from all the white, one from the red. This individual has one from the white parent, one from the red parent. This one too, from the white, and exactly. So all of these will have the same alleles. That's the only possibility, okay? So if you have one purebred crossed to another purebred, all their offspring are going to be heterozygous, okay? Another thing too, now next step that we're going to get into, when you actually take one of these individuals and you self it to itself, or you actually sib it, cross it to its sibling plant, okay? What is the possibilities? You can either only have white or red as the alleles from one individual, same thing from another. So what you do is you make a Punnett square. We're going to go into this in a little bit, so don't get all panicky, okay, at this point. So one from one from one, okay? This is one parent, yeah? We've got one allele, the other allele. This one here has one allele and the other allele. This is actually representing the kind of gametes that this parent is contributing. This is representing the gametes or the alleles in the gametes of this one contributing. So you actually bring this down, this one over. So this is a homozygous dominant. You know it's dominant because all the F1 look like this parent. Okay, so red in this case, red is a dominant factor. Okay, white is a recessive factor, the one that is not expressed in the F1. Okay, this one here is a heterozygote. You have R and you have W. Okay, so this is heterozygous, but it still has the red look to it. It's actually red. Okay, now this one here also has one red, one white allele, and this one also is red. So you have red, 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 and here is homozygous recessive. It actually has the traits that were masked in the F1, but it's expressed in the F2 because you have one and another, okay, of the same allele. So this one is going to be white. So this is the result you get. You're going to have a 1 homozygous dominant, okay, 1 RR, okay, to 2 RW to 1 WW. And that's going to be the genotypic ratio or the genetic makeup of the F2. In terms of the phenotypic ratio, the phenotype is actually what it looks like. You're going to have a 3-to-1 ratio. Okay, so don't worry, we're going to go through this again. Okay, so this is just introducing you to the idea. Okay, so as you just saw, the dominant allele is an allele that is expressed in the F1. So in the case that we just looked at, you cross a white flower colored to a red flower color. In the F1, all the F1 were red, right? So that is actually the dominant trait. The recessive trait is the allele that is not expressed in the F1. So the recessive trait in that instance was white color, okay? But this recessive trait will reappear in the F2. So Mendel, what did he think about this? Because this is the kind of results he got. Being, having his, pretty extensive background in mathematics, he figured, well, in flowers, in different kinds of organisms, there must be two. There must be two sets of these traits. He called them factors. There must be two factors, okay, for every trait in an individual. And this is generally borne out in most cases, okay? Now, so the next thing is that we have the genotype. That is a genetic makeup. That's actually the kind of alleles, okay, that the organism has. And usually we'll represent this with letters, okay, as we're going to see when we do a genetics problem, just like the one that we did, just like the one we kind of ran through a little bit before. Now, the phenotype is the expression of the genotype. It's what the organism either looks like or what kind of chemical it makes, okay, depending on what part of the body or anything like that. So the genotype is a genetic makeup, usually expressed in symbols such as letters, and the phenotype is what it looks like, okay, what it looks like or what is expressed. And the phenotype is based on the genotype. It's the expression of the genotype, okay? Okay, now Mendel kind of figured this out. This is one of his, one of the things he came up with, one of the ideas. He called it his principle of segregation, okay? So what happens is that these factors, which we call these factors genes, but he called them factors, okay? What happens is that these pairs of factors will separate, okay, during meiosis, right? Pairs of factors will separate during meiosis. So the sex cells, egg and sperm, okay, has only one factor. So it only gets one factor, okay, from the person who's actually making the sex cell, or from the plant that's making the sex cell, okay? So each sex cell has only one factor, okay? So remember when we were talking about alternation of generations, you have a little plant called the gametophyte that produces gametes, right? Where the sporophyte produces the spores and the gametophytes produce the gametes, okay? Next, okay, we're going to do something called a Punnett square. And in this Punnett square, it tracks alleles. Okay, so just remember, the principle of segregation has to do with the fact that the pairs of factors separate. So remember, Mendel was working in a vacuum at this time. He was actually doing this on his own. He probably talked to a lot of his friends, mathematicians and etc., but he was doing this all on his own. Remember, they didn't know about chromosomes, they didn't know about mitosis and meiosis, but he figured this out mathematically, okay, in terms of probability. Okay, so what we use is a tool called a Punnett square. We're going to see who Punnett was, Mr. Punnett was, Dr. Punnett was, later on, maybe in the next session. And we use a Punnett square. Geneticists still use this when they're doing their crosses. And what they do is they track alleles from one generation to the next. Okay, so next what we're going to do is we're going to get into how to do a genetics problem. Okay, so if we can go to the Elmo. Okay, so say, for example, in a genetics problem, I'll say, okay, we have two parents we're starting with, all right? One is, like in the case of this one, you can say one is homozygous or purebred for purple flowers. The other one is purebred for white flower colors. So in this case, you cross a purple one with a white one, all the offspring are purple. So from this, you know, you have information that the purple is dominant. Okay, so generally you assign symbols this way, capital, capital. This is the two sets of alleles in the purebred purple, and then you'll have lowercase, and so this is purple, and then you'll have lowercase p being white. You know that the white is recessive because all the offspring after this cross, the F1, are all purple. Okay. The next part you're going to do is you're going to do a Punnett square. So you have a purebred purple, purebred white. When the purple individual undergoes meiosis, the only possibilities in terms of alleles is purple, capital P's, right? When the white flowered individual undergoes meiosis, the only possibilities are white, signified by lowercase p's. I know it seems funny to give them, give white a P instead, but this is important because if you have P and W and you're tracking 3 or 4 sets of alleles, it can get to be pretty hairy, so to speak, okay? Anyway, to do a Punnett square, you do a little, so you have these kinds of alleles for one type of gamete, these kind of alleles from the other, because these are actually purebreds. So to make a Punnett square, you just have to make a little square thing like this. Okay, one parent along this way, the other parent along this way. The possibilities, the different kind of alleles they'll come up with. So you bring this one down, this one over, this one down, this one over, this one down, and etc. Okay? So therefore, these little 3 dots mean therefore. Okay? Therefore, all the F1, okay, have this kind of genotype. Okay, so that is sort of like the genotypic ratio. All of it, or 100%, have capital P, lowercase p genotype. Remember, genotype is generally represented by symbol. And all F1 have purple flowers. Okay, flowered phenotype. Okay, that's the first part. That's actually the first part of the genetics problem, okay? Now next, okay, next you need to do and try to figure out and predict what you're going to get in the F2, okay, in the F2 generation. So remember, when you have the F2 generation, you're crossing two F1s to each other, or you're selfing a single F1. Now when you have this F1, okay, you will actually, the two possibilities in terms of alleles for the gametes are capital P and lowercase p, okay? So next we're going to do a Punnett square for the F2, okay? So the same thing, you make this little, okay, like this, capital P, lowercase p. Remember, so each individual has two factors. One of them will be, since this is a heterozygote, you'll have 1 and 1. The other one will be the same, okay? Bring this down, bring this over to capital, bring this down, bring this over. I'm going in sort of like in a different direction. Bring this over, bring this down. And by conventions, you'll put the dominant trait first, okay? Lowercase, lowercase. Therefore, okay, F2, the genotypic ratio, excuse me, the genotypic ratio, so you have 1, okay, capital P, 2, this is showing ratios, 2, capital P, lowercase p, 1 and 2 to 1, okay? And this is the genotypic ratio in the F2. And then what you're going to look at this, you see that you have 3 that have capital letters, so you have 3 purple flowered to 1 white, okay? And this is the phenotypic ratio. All right? Oops, I'm writing off the screen here, sorry. Let's see, okay? So this is how I want you to do a genetics problem, okay? So let's see, I think we want to try to do another one, okay, just in case, just to make it perfectly clear, as an infamous person once said, okay? Why don't we do the seeds? So you can see that these are actually the same kind of crosses that Mendel did, okay? All right? Okay, so in this instance, we have smooth seeds and wrinkled seeds. You have one purebred for smooth seeds, one purebred for wrinkled seeds. You cross these two parents together and you get smooth seeds, okay? So you're going to name the trait according to, okay, first of all, you're going to actually make a key. You're going to actually name the trait. So smooth is dominant. So then you're going to name it S, okay? Capital S for smooth. You're going to have a lowercase s for wrinkled. I wish I had nicer handwriting. I never have had good handwriting. I've always had terrible handwriting. I'm writing neat for me. Okay? Okay, so smooth, wrinkled, purebred. So you're going to cross this and cross that. The only possibility in terms of alleles that the smooth one can come up with would be smooth, capital S, capital S. The only possibility that this individual can come up with would be lowercase s, right? In terms of the symbols that you're going to use. Only wrinkled alleles that it's going to pass on to its offspring. Only smooth alleles, this one will pass on to its offspring. So to show how this comes about, how you actually get all those smooth-seeded F1s, okay? You have capital, lowercase, capital, lowercase, okay? So this one and that one. Therefore, okay, all F1, okay, are this kind of genotype. Okay, uppercase S, lowercase s. They're heterozygous, right? Heterozygous. And all F1 are smooth-seeded, have smooth seeds. Okay? Okay, that's part 1. So you're actually showing how the F1 comes about. Okay. Next thing you're going to do is try to figure out, what do you expect in the F2, okay, the second filial generation. So this individual is a heterozygote, so this is the possibilities in terms of the alleles that they can donate to the F2, okay, for all these individuals, all these sister plants. Okay, and another thing to remember is that even though we put a Punnett square, because we have 2 factors, that doesn't mean that you're only going to have 4 offspring. Sometimes you'll have hundreds, sometimes you'll have dozens, depending on the situation. We're just getting a ratio, proportion from one to the other, okay? Anyway, so we'll set up another Punnett square here, okay? Capital, lowercase, okay? And here's one, here's another, here's another, and then here's another. Yep. Capital, 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 lowercase, capital, lowercase. Okay. Therefore, okay, you have 1 capital S to 2, right? 1 capital S to 2 capital S lowercase s to 1 lowercase s, and that is your genotypic ratio, the genetic makeup, okay? And then for the phenotypic ratio, you have this one, and this one with capital letters, so you know that's a dominant trait, so then you know that you're going to have 3, let's see, smooth-seeded individuals to every 1 wrinkled-seeded individual. Okay? All right, so that's pretty much what I want you to do for your genetics problem. Okay? Okay, all righty. Okay, let's see. Then what we're going to do next, okay, we're going to actually give you your genetics problem to work on. Okay, so I'm going to leave it on kind of long time so you can copy it. Okay? Now, first of all, you start off with 2 purebred plants. Okay, you'll have 2 purebreds. Remember, so what that means is that both parents are homozygous for the traits. Okay, one has the genes or is homozygous for yellow seeds. The other one is homozygous for green seeds. Okay, you cross-pollinate these plants. That means you take pollen from one plant, put it on the stigma of the other plant, and then so you actually cross 2 plants that have this one trait different between them. Okay, this is a monohybrid cross. Only one trait differs between the 2. You cross-pollinate these plants, and all the offspring that come out, all the F1, have yellow seeds. Okay, so you have 2 purebred plants, one with yellow seeds, the other with green. You cross-pollinate these 2 plants, all their offspring have yellow seeds. Okay? Is that enough time? Okay? Is that enough time, you think? Alrighty. Secondly, the first thing you need to do is you need to assign symbols for the P generation, for the parents. You need to assign symbols for the P generation. Usually you'll name after the allele, okay, the allele that's dominant. Okay? So you assign symbols for the parental generation. Then next, you should construct a Punnett square to show, okay, how the F1 came to be the way they are. Okay, remember the F1 all have yellow seeds? So actually make a Punnett square, construct a Punnett square to show how the F1 came about. Okay, what actually went into the making of the F1, or the first filial generation. Okay? Next, answer this question. What is the genotypic ratio of the F1? Okay, what is the genotypic ratio of the F1, or the genetic makeup of the F1? What is the phenotypic ratio of the F1? What do they look like? Okay? That's part 1 of this problem. Alrighty? Next, second part. Construct another Punnett square to show what you can expect in the F2 generation, or the second filial generation. Okay? What is the genotypic ratio of the F2? Okay, you must actually give me the numbers as well as the symbols. Okay, you need to actually give me numbers and symbols. What is the genotypic ratio of the F2? What is the phenotypic ratio? Okay, yeah, easy, yeah. Okay, what I hope to do is that I hope to put this on the WebCT, so you should be able to get the problem on the WebCT as well. Okay, it's a good idea to start work on this. Okay. So you'll have 1 week to turn this in. 1 week from this date of assignment until you need to turn it in. Okay? All right? All righty. Okie doke. Let's see. Next, so we're still on the monohybrid crosses. Okay? So another kind of monohybrid cross, or actually monohybrid crosses has to do with differences between, one difference between 2 different plants. We also use these crosses to do something called a test cross. Okay, what is a test cross? A test cross is when you take an organism that has either that generally has a dominant trait looking. It looks like the dominant type. So in the case of purple flower color, it would have purple flowers. But if you don't know what the genetic makeup is, what you can do is you can cross this purple flowered plant to a white flowered pea plant. So we're talking about pea plants right now. Because if the purple flowered plant is homozygous purple, if it has both alleles for purple flower color, you cross it to a recessive, right, a homozygous recessive. If all their offspring are purple, okay, you know that the purple flowered one is a purebred for purple. If half the offspring are purple, half the offspring are white, right? Because if you have one allele for purple, big P, one allele for white, little p, okay, or lowercase, then half of them are going to be purple, the other half are going to be white. That's something called a test cross. That's the way you can kind of figure the genealogy, or you can actually figure the genetic makeup, okay, of the plant that you don't know what it is, even though it expresses the purple flower color, okay? Another thing too is that people also do dihybrid crosses. Yikes, okay? So what they'll do is they'll actually have two sets of traits that they cross through, that they actually follow through several generations. And I believe Mendel, let's see. Anyway, so some people do these dihybrid crosses, okay? Sometimes they do trihybrid crosses, or even, you know, tetrahybrid crosses, I guess they would call them. Now these hybrid crosses are actually still used, you know, people still use the Punnett square. It is very useful because geneticists, probably even at UH and in the biomedical building, you know, in the medical field, I forgot what you call that. Yeah, you have genetics. You have a discipline known as genetics. You can even major in genetics in some places, right? But they actually do have, you know, Punnett squares up on the board where they're actually tracking 2 or 3 sets of traits through a couple of generations. People still use the Punnett square. So this is very good for you to practice with, okay? As well as being your second learning log, okay? So here we have showing a dihybrid cross using these rabbits, all right? So you can see you have a white rabbit here. This is actually white, and this, so it has lowercase b because it's white. This one has uppercase b because this is a black rabbit. Okay. Another thing, I believe that this one here is short-haired. Okay, I believe SS stands for short-haired, uppercase S, which is dominant, and this one here has long hair, which is little s. I believe that's what it is. When these two rabbits are bred together, okay, you have this organism, you know, uppercase S, lowercase b, and etc., crossed with a lowercase s, uppercase b. What you'll have a whole bunch. They're being rabbits too, right? Of rabbits here. Okay. Anyway, so you have them like this, right? Anyway, just showing that when you're tracking two sets of traits, that generally what you'll tend to do is you'll put them in these boxes. You can still do a Punnett square with that. And one of the things you'll notice about this kind of Punnett square, that generally you're going to have a 9. You can see that this one here, okay, has 9 to 3 to 3 to 1. Okay, what you'll have is a 3-to-1 ratio inside of each other. Okay, 9, okay, to 3 to 3 to 1. That's what you usually expect from what they call a dihybrid cross. Okay. Anyway, that's kind of interesting. Yeah, so people do dihybrid crosses, they do trihybrid crosses, they do tetrahybrid crosses. You know, they'll actually track several traits over several generations sometimes. It can really get kind of hairy, so to speak. Yeah, right, we're talking about rabbits. That was a joke. Okay, now beyond Mendel, okay, you have something called incomplete dominance. So this is not something that happened when Mendel was doing his pea plant experiments. This is incomplete dominance. That means neither trait, when you cross two different purebreds together, neither trait is dominant. Okay. So this is one. This is a very, very good example. So here, you know, using the same example of a parent with white, you know, alleles for purebred for white, the other one purebred for red. Okay, you cross them together, but all their offspring are pink. Isn't that amazing? Right? So what you'll have is you'll have an intermediate phenotype because neither of the parents in this case are completely dominant. But when you actually do, you know, the next generation, when you self the F1 or you sib the F1, crossing sister plants to each other or selfing one of these plants, what happens is that these factors will actually segregate into this way. You'll have 1 red to 2 pink, right? 1 homozygous, okay, to 2 heterozygous to 1 homozygous. Okay, so this is an example of incomplete dominance. Okay, so that happens in Okay, so the F1 has an intermediate phenotype, and then you also have that the fact that the phenotypic ratio and the genotypic ratio are the same. Okay, and this happens in plants known as snapdragons. A lot of times in Hawaii, we don't really see snapdragons just because they're more like a temperate area plant. But if you go to a good florist in the springtime, they can get you snapdragons if you like snapdragons, you know. Now we also have some things, you know, some examples of what they call multiple alleles. All right, multiple alleles. So when that happens, you have more than 2 alleles in a population exist for any particular trait. A very well-known one are the ABO blood types in humans, okay? Because ABO blood typing is one way that people can figure out, you know, what kind of blood somebody needs if they need a transfusion or etc. Okay? So this here is like a depiction of a red blood cell, an erythrocyte, and this one is supposedly an erythrocyte or blood from somebody with type O. In the past, in the olden days, they used to call type O people universal donors, okay? Because somebody with type O, you know, O actually stands for 0. That means it lacks certain proteins that are presented on the outside of the cell. People who have A proteins, what they call A antigens, have A-type blood. People who actually have B-type, no, not antigens, yeah, antigens. So they'll have A-type blood, they have the protein, A-type protein. People with B blood have the B-type protein, but the ones with O blood doesn't have either one. So somebody with O blood, everything being equal and all the other blood typing being taken care of, can donate to people with type A blood and to people with type B blood. But A cannot donate to B, B cannot donate to A. Okay, because they'll actually have immune response, stimulating immune response and tissue rejection, you know. Furthermore, people with A-type blood, everything being equal, the RH, you know, positive and negative things and other ways of blood testing, A blood people can give to somebody with AB type blood. People with B-type blood can also give blood to AB type, and O-type blood people can also donate to AB people with AB blood type. Now, AB blood type used to be called the universal acceptor because it could accept blood, all things being equal, everything, you know, being okay with RH, you know, factor and everything too. Everybody could donate blood, okay, to this universal recipient. Okay, that's like in the old days. So you have these multiple alleles. Another example of multiple alleles are clover. Okay, leaves of clover. Here's some clover. Okay. You can see on some of these, you actually have these striations or markings on them. Okay, the presence or the absence of markings on clover leaves actually has to do with several different alleles. Okay, another kind of clover here too, right, as well. All right, with this kind of marking. So the markings on these different kinds of clover, okay, are actually coded for, okay, by different sets of alleles. Okay, so there are more actual sets of alleles of these that exist in the population. Okay, more than 2 sets. We also have another kind of something that comes up called polygenic inheritance. This happens when you have a trait controlled by more than 1 pair of genes. Okay, this is a trait that's controlled by maybe 2 pairs of genes, 3 pairs of genes, 4 pairs of genes, right? Sounds like something out of somebody's closet, huh? So skin color in humans, okay, is actually a trait that is actually controlled by more than 1 pair of genes. It's kind of interesting. So here, this is a map of the world showing people with really, really fair skin, okay, on this part, right? They're all kind of clustered here, like really, really fair. Okay, so they actually have the scale here. Then you have really, really dark-skinned people all clustered over here, okay? So you can see that a lot of the people in the world are kind of intermediate. So skin color in humans, pretty much people think, is actually determined by more than 1 set of genes. I think some people say that there might be 6 or something, 6 pairs of genes, 4 pairs of genes, okay, in humans, all right? So that can happen, and that happens too, you know, and that sometimes you can have 2 really, you know, dark-skinned people, and sometimes your child is fairer than either of them, you know, like that. That happens, okay? Another example of polygenic inheritance is the seed color in wheat. So this is just some wheat plants growing, probably in a botanical garden or in an agricultural station, because it's labeled. You saw the plant label there. Now, more things happen, is that these 2 fellows, you know, very serious-looking guys, yeah, William Bateson and Reginald Punnett. This is the Reginald Punnett that invented the Punnett square, famous fellow, yeah, in 1906. Okay, 100 years ago, they actually figured out something called linkage. Okay, what is linkage? Okay, this is when genes on a single chromosome are inherited together. Okay, so they actually, so genes on one chromosome, since they're on the same chromosome, okay, are inherited together. Okay, so you didn't have the usual segregation like you saw in Mendelian genetics, or genetics as done similar to Mendel. Okay, so they studied the sweet peas. This is a different plant from the pea plants that we talked about earlier. These are called sweet peas because the flower is supposed to have a sweet smell. Okay, and they're temperate area flowers. I think they have a quick germination time and growth time. You can actually grow several generations just in a few months. So sweet peas, another group of plants. Well, what they had is they actually had, you know, purple sweet peas and red sweet peas, and they had long pollen and short pollen. That means the pollen was longer, or sort of like longer, ellipsoidal or something. So what they found out is generally these two traits, purple flower color and long pollen, were inherited together, or there was like a much larger, you didn't get the 9:3 to 3:1 that you would get in a usual dihybrid cross. In fact, this is also in your textbook, okay, where you have the parents being purple flower, long pollen, red flower, round pollen, like you see here. Okay, the possible gametes. So what they had is they had a whole bunch that were purple and long and red and short, and not so many that was purple and short and red and long. Okay, just because they found out that these genes tended to be inherited together. So what could account for the fact that you had the recombinants? You had long and red and short and purple. Okay, we're going to get into that in just a sec. Okay, when we get into Thomas Hunt Morgan, he also did work in the early 1900s, very important in terms of his genetics work, one of the towering giants of genetics. Okay, so crossing over, remember crossing over from meiosis? What happens? Okay, what happens? When does this happen? I already told you, meiosis. Okay, so what happens? You actually have chromosomes. You remember homologous chromosomes? Even if one is black and one is white here, they're still homologous chromosomes. They actually color them different to make so you can see them better. What happens? You have crossing over. You have one chromosome and another chromosome that are homologous. They're attracted to each other. What happens is that they both break and they exchange segments. Crossing over. This is a way to do genetic recombination. So when Thomas Hunt Morgan did his work, that really could explain what happened with Bateson and Punnett. You know, that they actually had these recombinants. They had the red flower with long pollen and purple flower with short pollen. That could happen even though the two genes were on the same chromosome. You know, the two different alleles for flower color and for length of pollen, they were on the same chromosome, but the chromosome can actually break and exchange parts, okay, with its homolog or homologous chromosome. Okay, isn't that amazing? Yeah? So way back in the early 1900s, he actually rediscovered Mendel, gave him the credit that was due him, even though he had already passed away by then. But still, you know, I think people, especially in the religious life, they really do know that it doesn't matter who does it. It's just that the work gets done. Here's another one that shows, you know, these two homologous chromosomes where you have crossing, double crossover. Okay, so you can actually have chromosomes recombining. So this is another reason why we humans and plants and stuff, why we're so variable. Because due to sexual reproduction, due to crossing over happening during sexual reproduction. Okay? Another example, so this is just giving you a little bit of these, this is actually adenine, guanine, cytosine, and etc. So here, this is actually from the male parent. This is from the female parent, okay, from the female parent of the organism that has this chromosome. Okay, so what happened is that you have this crossover event. The chromosomes break and they exchange segments. So this one here this is a recombinant. It has genes from the father and from the mother on the same chromosome. Same here. You have genes from the mother and on the father. I don't know whether that was male or female or mother and father. I guess it must be mother and father, red and blue, you know. It's kind of interesting because generally in the East, in the Eastern world, red is more like a masculine color and blue is considered to be a feminine color, yeah? The yin and the yang, yeah? Or the yang and the yin. Anyway, it's sort of like different ways of looking at the same thing. Okay, so what he did is he actually, Morgan too, actually used the right organism. He used a fruit fly called Drosophila melanogaster, okay? So here's this is the male, Okay, and this is a female. I know that we humans, we're kind of biased. We always tend to assume that because, in our species, males tend to be larger than females, but in other species, sometimes the females are larger. Okay, so in this case, the female is larger. Okay, another thing too, this also happens in other kind of animals. Like the raptors, like eagles and hawks and falcons. Generally, they too, the female tends to be larger than the male in that case too. So we humans, we definitely have our biases, So even if you have children's cartoon, and you have insects or you have birds or anything else, generally the male one will be the larger one, But that isn't always the case, not in nature, okay? All right, anyway, in order to summarize, so we covered a lot of ground today, but now we're going to summarize a little bit of what we've done. I know it's a lot, okay, but you will learn this thing, okay? First of all, we went through a little bit about the principles of inheritance, okay? We talked about the father of genetics, Gregor Mendel. Amazing, amazing man, just amazing. And we also talked about Mendelian genetics, what we call classical genetics, as well as some of its offshoots and developments. And we also talked about other patterns of inheritance. Okay, all right. So that was a lot, right? That was a lot to cover. I know we went through it pretty quickly, okay? Be always sure to email me if you have any kind of questions. And I guess I'll see you next time. Aloha.